everyone. I am Adelaida Turku, and with the two Alexes, we're going to present. We are members of the EEG team. So who are we? Our vision is basically to understand the correlation between the data analysis, the quantifiable data, and the psychological presentation. We basically, and then the mission, it's of course like the wellness of our participants. We want them to feel secure and safe, but also to understand the psychophysiology and of course like measure the data, analyze them, see what we're gonna go, where we're gonna go, and then also we want to execute to give also the students, our members, the opportunity to do some research. Meaningful research. And the science behind what we're doing right now is that we want to, through the equipment that we have, that we are given from the school, we want to understand how to stimulate the parasympathetic nervous system. How does that work? Through the ISF trainings, we want to, with the sound therapy, arrive to the brain and then simulate that system that gives you, relaxes you, gives you that calm and that kind of feelings, and then shut down the stress and the anxiety. So our values are, of course, the wellness of our participants. We want them to feel fine, and then our members, they're not only given the chance to be trained to use the equipment, but they also are required to do NIH certification, which is related to human subjects research. And then here we want to be engaged in research using the scientific method, as you all know, because we are all in STEM. Observation, and then hypothesis, experiments, test, verify the hypothesis, and then go back. So that was very useful for our ISF trainings when we had to understand what was the optimal frequency, the one at which our brain relaxes. And since each of us, each participant, is very different, they have different biology and different physiology, so that we had to apply our scientific method. And it was very, it was like a practical moment where you could understand a theory, like such a concept into in a field. One more thing. So our goals are basically to gather more participants, because it's not an invasive method. So please, we're gonna go and share your contact information if you're interested in it. It's non-invasive and very. If you have like other questions, more questions, we are more than happy to answer to you. Personally, I'm gonna, once we get the keys from the company, it's gonna be very soon, we hope, and we're gonna start running the trainings. And personally, I'm gonna be doing that because Professor Di Loro did that last year. She felt amazing. Her parasympathetic nervous system worked incredibly fine. And then we want to share another story. Can you please? Yes. We want to share our spring story, which is a successful story. We had a veteran last semester. This is the QEEG, which is about basically our post. After all those trainings, we want to record, we want to see the brain activity. And this is like what we got. Those like are all the brain waves, okay? And then the red stripes here are the artifacts, like the parts that the QEEG, the equipment that we're using doesn't get. Anyways, our successful story was of this veteran who basically, before he was a PTSD, he had a traumatic experience at war, and before going there, he was an amazing athlete, going like 2 times a day to the gym, being very active. After the war, he was traumatized. He couldn't have that life anymore. He couldn't go even outside, just going to school, following a certain curriculum, and that's it. after those trainings, he was able to go to the gym again, at least one day, once a day, and every day. So please feel free to contact us, and one more time, we're gonna be more than helpful, more than happy to answer to your questions. And now the equipment parts, Alex.
Hi. Actually, I'll be passing around the room some of our equipment. You know, some of them very expensive, some of them not so much. And passing around the actual the QEEG cap that we use during actual study. This is quite expensive, but you know, it's just seriously numbers. And actually,
don't throw it on the ground.
On the, I guess, on my right side, you have the cap that we use for the QEEGs, which connects to the actual DC amplifier on the other side of the room. Allows us to record the electrical, let me see, the voltage of each area of the brain. We use a saline solution, which is injected into the ports of the 19-channel system. That DC amplifier doubles to components. It has a QEEG section, and it actually has an ISF. In the back, they actually have the other electrodes. It's the superchloride electrodes. Those were actually, I think, a five electrode system where you actually put on your mastoids, your references, your T3, T4, and the actual ground, which you place usually either on the neck or on the forehead. Let me see, the cost of each equipment, I think the electrodes in the back are $130. The actual equipment over there on the other side should be around $6,000. And combining with the actual BrainMaster software, which we're still waiting on the password keys for, actually allows us to do our research through the combination between our network system or local network and the actual equipment. We're actually able to not only record it, actually do the experiments, which will be the actual ISF sessions, which is, I think, 6 weeks. 2 times a week we'll be doing our sessions, which will last for 30 minutes long. We'll be asking questions during the, let's see, with the length of the 30 minutes, we'll be initially asking with the QEEG, and then the actual, let me see, the GAT7 forms, which will be the next room. Yes. Yeah. And actually, that's kind of happened so far. This is Dr. Delora. So here's Tyler and our EEG member Anthony actually setting it up. We actually had to go through a training with Delora shows a video for and placements, and we actually had another device in the room, the Checktrode, just in case actually the DC amplifier has an impedance, allows us to check if the connectivity is actually working for the placement of the gel.
Yeah. So I'm going to talk to you about kind of what we've done for the past couple of weeks. Part of our goal, our first goal, was to set up the EEG project for the fall semester. One of the biggest difficulties is that when we get new researchers, actually members of our project, not participants, it's kind of hard to kind of onboard them and allow them to kind of flourish by themselves to collect data without supervision. And obviously, if we can create independent researchers that can gather data without outside help, then we can really explode kind of as far as how much data we're collecting and hopefully get a statistically significant N. So that's kind of the first goal, was to establish those methods. I'm standing in front of the text here. This is just some text from an example of one of the tutorials we have. Again, over there is some of the information, or I'm sorry, some of the hardware that we have that you've got your hands on. Our second goal was to, again, recruit participants, as we've been doing, sending things around. And the third was to create new futures for the EEG project, because we've kind of been singing the ISF tune for some time now, and we went kind of back to our roots. We had 2 pieces of hardware / software. One is called the BioPack, which allows us to do all sorts of interesting things with polygraphs and blood pressure measures, pulse, and even like a switch button like you'd see on a game show, which we could use for things like reaction time. The OpenBCI is a more malleable interface than the Brain Avatar, which is the software that we use for that thing we've been passing around. And we hope to eventually integrate measurements we take from the OpenBCI into other projects, or even we were throwing around ideas about thought translation, which can hopefully take inputs directly from the mind to as close to machine code as possible, pretty much. So is there another slide here? Yeah, so I probably already talked about some of this. The OpenBCI cap is already wireless, but it's kind of clunky, and we were considering maybe if we could put our minds to a better design and something more portable, a little less brittle. Again, the OpenBCI allows us to have that flexibility. It'll be more compatible with outside software. And again, the BioPack, I talked about those. And so up here is our current meeting times and all our pertinent information. You'll see the emails here at the bottom. It's just our Bergen emails, pretty much. You can see us in the dental hygiene clinic. You probably have. We're kind of there all the time. And I don't know, I kind of wanted to pull up the, since we made all our forms.